My name is Dr. Greg Schneider with Applied Cells, and today we are going to talk to you about a multiphysics approach for complete cell preparation and isolation using Mars. Today, our panel is Nick Wilson, a product manager at Applied Cells, and myself, a technical sales specialist with Applied Cells. And we will be talking about the approach we take at Applied Cells with Mars for cell preparation and isolation, supporting tumor biology and immunotherapeutics. To introduce this Mars system, I will show you a short video. The Mars system, as you saw in the video, is divided into three modules. The first module on the left here is the cell wash module. The center module is the cell concentration module. And the rightmost module seen pictured here is the immunomagnetic module. The cell wash and the cell concentration module use a unique acoustic chip technology, and they can be used in conformation where the cell module runs into the cell concentration module in a serial fashion to get a wash and concentration. Alternatively, it can be run in the conformation where both these first two modules are cell wash modules to increase throughput with two different samples. The third module, the immunomagnetic module, is a unique proprietary immunomagnetic module that allows the capture of cells with specific biomarker phenotypes. And we will be discussing all three of these modules in upcoming slides. Before, the MARS, which stands for the Multiphysics Automated Reconfigurable Separation, has two proprietary technologies. The first is our CPC cell processing chip, which is based on acoustic cell manipulation. The input of the input sample is seen here as our debris stream. And as the acoustics are tuned, the cells of interest are forced from the laminar flow of the debris stream into the cellular or sample stream, and therefore captured. For the immunomagnetic separation, the cells are labeled with immunoglobulins for specific biomarkers and then run through our system. The system is a flow-through, matrix-free system that is gentle on the cells, and it has the benefit of all of the cells that go into either the positive or negative fractions are completely recoverable. Therefore, it makes negative selections much simpler. As you see here, the system comes in two basic conformations: the Mars sample prep, which allows parallel sample processing, and it has three of the cell wash and concentration modules that can be conformed to run up to 3 different samples in parallel. The Mars cell separation configuration of the system is the one that has the immunomagnetic separation module as the third component of the system. Both these systems are designed to be fully automated and walk away. And with that, I will hand it over to my colleague, Nick.
Thanks, Greg. As you can see, Mars enables a wide range of versatile applications that can be integrated into nearly any cell-based workflow. The first and most obvious application of this technology lies in its ability to effectively process and wash samples without the need for centrifugation. The acoustic microfluidics cell processing chip, or CPC, is integrated into both configurations of our Mars systems and can be used to automate wash and sample preparation for effective characterization of cell populations via flow cytometry and imaging analysis. By fully automating this sample preparation workflow, the Mars enables consistent and reproducible results, achieving high recovery with superior debris removal, thus leading to high-quality downstream analysis. Building upon the acoustic cell processing technology, the Mars also offers magnetic separation module. By combining these two separate technologies into one complete system, Mars provides a front-end solution for any cell-based assays, including the detection and enrichment of rare cells from whole blood, such as CTCs and DTCs, with high recovery, high purity, and high throughput capabilities for downstream genomic or transcriptomic analysis, yielding high-quality data with minimal noise. And finally, these combined technologies can also be used to automate and streamline immune cell isolation workflows, whether it be aimed toward endpoint immune cell profiling and analysis, or for continued culture of isolated immune cell populations. The Mars system was initially created in order to address and improve upon the long and tedious nature of the industry standard workflows aimed at enriching immune cell populations from whole blood. I think anyone who has experience with FICAL prep would agree with me that it is one of the most painstakingly long workflows that is prone to high variability due to unavoidable manual operating error. Additionally, white blood cell enrichment through red blood cell lysis and centrifugation of whole blood commonly leads to some degree of cell loss through each centrifugation step. Thus, as you can see, the Mars provides an effective solution to each of these pain points, as it provides a simple, automated workflow that can yield high recovery while nearly eliminating all manual hands-on operation. With a sample flow rate of about 1 milliliter per minute, these workflows can be achieved at high volume and high throughput, cutting down total runtime significantly. As you can see by the pictures located on the top left, the process is quite simple. You add your red blood cell lysing buffer to your whole blood sample, run it through the optimized Mars wash, and the result is 2 outputs: your enriched white blood cells, as you can see on the left, and your waste collection, which includes all of the washed out red blood cells, platelets, and debris. In order to confirm that the Mars system does not buy a sample recovery, we conducted flow cytometry analysis to characterize our white blood cell and lymphocyte subpopulations. As you can see from the bottom 2 figures, immune cell subpopulation characteristics are nearly identical when comparing the Mars to the original lysed blood sample, as well as conventional centrifuge wash. You will also notice that not only did the Mars wash not buy a sample recovery, but it also achieved significantly higher cell recovery of the various subpopulations compared to the standard wash protocol. Another important thing to note is that the continuous sample flow of Mars's microfluidic platform greatly reduces cell-to-cell contact and any unwanted activation or stimulation of your cells, as it does not require any pelleting. Next, we wanted to test the efficiency of this system's debris removal. We started by running flow analysis on our original lysed blood sample before any of the washing steps, and then compared Mars wash to standard centrifuge washes. On the left, you will notice a much lower amount of residual debris in the Mars wash sample output. And on the right, you will see that Mars was able to achieve greater than 99% red blood cell and platelet removal while maintaining a greater than 90% cell recovery. Next, we wanted to test the system's sensitivity and range by conducting recovery experiments at varying input volumes and cell concentrations. The graph to the left shows the results from testing the lower threshold of sample input volumes, in which we tested volumes of 200 microliters all the way down to about 10 microliters. You can see that even at 10 microliters, we were able to achieve over an 80% recovery, with the recovery rate increasing as input volume increases. On the right, you will see that we conducted experiments to test Mars's separation efficiency at varying sample concentrations. Results confirm that we were able to achieve good cell recovery and linearity over 6 orders of magnitude in cell concentrations. Now, in order to put this system's high throughput capabilities to the test, we ran 60 milliliters of lysed whole blood continuously over the course of 1 hour, analyzing sample outputs every 15 minutes, identified by 1, 2, 3, and 4. As you can see, even in long-term continuous operation, we were able to maintain greater than 90% recovery over each time period, thus illustrating Mars's stability over long-term operation. Now, moving beyond the simple whole blood processing, we wanted to see how the Mars system was able to perform when processing a solid lung tumor biopsy. We first obtained a baseline through our original lice no-wash sample, which had been stained for white blood cells and tumor cells via CD45 and EpCAM. We then ran that sample through both centrifuge wash and Mars wash. And as you can see, Mars was able to achieve high recovery while also drastically reducing the amount of debris and dead cells in its sample output as compared to standard protocols. Building off of this previous experiment, we wanted to test the tunability of the Mars acoustics in achieving various output goals. The tunability of the system lies in its ability to adjust the voltage of our acoustic frequency. We often crank up the voltage when we are focused on achieving high recovery and minimal cell loss, while we can also reduce the voltage to achieve a cleaner sample output with higher purity and less debris. You can see the results on the right compared to centrifuge wash. With high recovery, with high voltage, and high purity with low voltage. You may have noticed that the low voltage sample did not achieve a very high TIL recovery, which makes sense as the settings were optimized for tumor cell enrichment rather than lymphocyte enrichment, which are comparably smaller in size and density. But this brings up a key point to remember about this system. Throughout the wash process, no cells are actually lost. All sample inputs either come out in the positive fraction, which is typically the target cell output, or the negative fraction, the waste output. And both can be further evaluated based on your specific workflow needs. Thus, this low TIL recovery only represents the TILs found in the positive fraction. What this system enables you to do is to run your first wash at low voltage to achieve high purity of tumor cells found in the positive fraction, but then you can also take the negative fraction in which our seemingly lost TILs are expected to reside and run it through a second wash at high voltage to recover any remaining cells in that waste fraction. As you can see, we were able to recover many of the TILs and tumor cells that were not originally covered in the first wash cycle, thus further illustrating the point that no cells are actually lost throughout the Mars wash process. Now, moving beyond the acoustic cell processing technology alone, we can also utilize our magnetic separation module for the detection and enrichment of rare cells such as CTCs and DTCs from whole blood in one complete workflow. As you can see, the rare cancer isolation workflow is quite simple. You start by labeling your blood sample and adding lysis buffer. You then run it through Mars wash in concentrate in order to remove any red blood cell debris and/or dead cells. Then add magnetic beads and run it through the magnetic separation module by performing a CD45 depletion in order to enrich for only your target cancer cells. Once again, you will notice little to no manual operation required throughout this process, giving you more time to tend to other things in the lab as the Mars does the work for you. In order to test this, we conducted several spiking experiments in which we spiked tumor cells at varying concentrations into whole blood and tested cell recovery at each step of the process. You will see that with an N of greater than 10, we were able to achieve greater than 95% target cell recovery through Mars wash, greater than 99% recovery through Mars concentrate, and about 95% depletion of white blood cells through Mars magnetic separation. And in the end, we achieved greater than 90% recovery of spiked tumor cells, even at varying spiking concentrations. Taking this proof of concept and applying it to more clinically relevant applications, we were lucky enough to partner with Dr. Beth Chislock at the University of Pennsylvania to study the detection and analysis of rare disseminated tumor cells commonly found in the bone marrow of breast cancer patients using the Mars system. To provide a little bit of background on Dr. Chislock's work, To Prevent standing for Secondary Prevention through Surveillance and Intervention, is a multidisciplinary translational research center that combines basic science and clinical research in order to study breast cancer recurrence through the detection and treatment of patients with minimal residual disease, with the end goal to reduce tumor recurrence in patients. Studies have shown that over 20% of breast cancer survivors will experience tumor recurrence at some point as far as 20 years after initial diagnosis, and that DTCs are detected in the bone marrow of over 30% of early-stage breast cancer patients. We also know that dormant DTCs exist in the bone marrow, and the presence of these DTCs represents a poor prognosis factor for breast cancer patients, as the renewed growth of bone marrow DTCs is ultimately what causes tumor recurrence. Thus, patients with detectable DTCs in their bone marrow are more likely to relapse and have decreased survival rates following initial diagnosis. Although detection of bone marrow DTCs can help identify breast cancer patients with increased risk of recurrence, there are currently no specific therapies targeted toward understanding the biology of these dormant DTCs. Since DTCs represent the pool of cells in which breast cancer recurrence can arise, having the ability to efficiently detect and eliminate DTCs could have a significant impact on clinical outcomes for the millions of breast cancer survivors who are at risk for tumor recurrence. Thus, there is an urgent need to improve the methodology in which we detect bone marrow DTCs, and this is where the MARS comes in. As you can see, the current methodology for detection of MRD and DTCs have several significant drawbacks. First off, standard workflows require extensive hands-on processing, which includes several centrifuge wash steps, each of which can lead to some degree of cell loss. In addition, you'll notice that the standard method is extremely long, and every additional hour of processing can cause your precious and fragile DTC samples to die. So reducing the number of steps and overall time is critical. With the MARS system, we are able to greatly reduce the complexity of this workflow, eliminate multiple processing steps, reduce hands-on sample manipulation, and reduce overall work to 1/4 of the time required by conventional methods. Dr. Chislak and her team were able to conduct extensive studies of DTC detection at varying spike frequencies to assess our system's performance. Although there is variable recovery at lower spiking frequencies, which is to be expected, the MARS system was able to consistently achieve 80 to 90% DTC recovery across a wide range of spiking frequencies. Most notably, they were able to successfully achieve DTC detection at a frequency as low as 1 in 5 million bone marrow cells, which is the lower threshold of observed DTC frequencies in breast cancer patients. We are all very excited at these results, and the folks at 2Prevent are continuing to conduct additional studies with the MARS system and plan to extend testing of these applications on clinical samples from breast cancer patients. And with that, I'll now hand it off to my colleague, Dr. Gregory Schneider, who will be discussing some of the immune cell applications and data that Applied Cells and some of our other partners have recently generated.
Thanks, Nick. I'm now going to talk about the use of the MARS system to isolate immune cells. The MARS system allows for higher recovery, purity, and accuracy of immune cell type isolation. The system automates the isolation step for immune cell isolation for downstream immunotherapy manipulation. The first steps in immune cell isolation are the same MARS treatment of whole blood that you've seen before. The lysed blood is applied to the first wash module to remove the lysed red blood cells and other cellular debris. The second module is an optional concentration step. This step is usually employed by researchers to reduce the volume, which can be very useful to reduce reagent costs. The final module seen here is the MARS magnetic separation module. The cells are labeled with biotinylated antibodies directed against specific biomarkers and then incubated with streptavidin magnetic beads. The MARS system is a continuous flow, matrix-free, gentle system without a traditional column that can trap cells. The cells that are put through the magnetic separation are recovered in the positive or negative fractions. This allows for very simple negative selection experimental design. As you see here, With the negative selection, we can do very powerful experiments isolating PBMCs or lymphocytes. The whole blood was Mars washed and labeled with CD45 to detect the immune cells. The cells were then incubated with either biotinylated CD15 or biotinylated CD14 and CD15. The cells were then incubated with applied cell streptavidin beads to remove granulocytes or granulocytes and monocytes. The results show that PBMCs were recovered in high purity and high recovery, and lymphocytes were also recovered in high purity and high recovery. This allows downstream study of these cells. And again, the negative selection of these cells makes it possible to study cells that have not been previously bound by antibodies and possibly activated. This schema shows the Mars workflow versus the conventional workflow. As you can see, the Mars workflow is greatly simplified and reduces time to results. Additionally, since the Mars system is fully automated, lab personnel are free to do other laboratory activities. This is a busy slide, but we wanted to show you all the data. So to guide you through this, the first column is labeled with anti-CD45 to indicate all hematopoietic cells. The second column is labeled with CD3 antibody, and this is going to be a different clone than the clone to capture CD3 positive cells. The third column is CD19 labeling the B cells, and CD15 labeling the granulocytes. The cells were Mars washed and incubated with biotinylated anti-CD3, again, a different clone than the CD3 labeling antibody, and incubated with Mars streptavidin magnetic beads. As you can see, with a single Mars experiment, you can generate highly pure samples of CD3 in excess of 90%. However, you can also track where the B cells are, where the granulocytes are in this experiment. Again, showing that the Mars system allows you to track your cells to either the positive or negative fractions with virtually no cell loss. A very powerful tool in assay development. Mars magnetic separation is also useful in a positive conformation. So here you see the input cells from the Mars wash here on the left side. The positive fraction using anti-CD8 antibodies binds and separates the CD8 positive cells with a very high purity and very high recovery with minimal CD4 positive cell contamination. The majority of the cells here are CD8 positive cells. The negative fraction shows you some cellular debris and the CD4 cells. The conclusion is that the Mars magnetic separation quickly purifies CD8 positive T cells, again for further downstream use for your research. This experiment shows a co-culture experiment that we recently had. This is generating purified CD4 positive cells from a negative selection experiment. And this is a key request that we often run into. The input cells show both the CD4 population here and the CD8 population. The cells, as I said previously, were from a co-culture experiment, so this is not from whole blood, but from cell culture medium. The cells were washed in our CPC and then labeled with anti-CD8 antibodies and incubated with our streptavidin magnetic beads. The sample was run on the Mars magnetic program with the positive and negative fractions collected and assayed. As you can see, the negative selection of CD4 positive cells is very pure, and it can be further studied in downstream applications. Placing the Mars system in a sterile cell culture hood allows the researcher to isolate immune cells that are immediately ready for culture and proliferation without ever having to leave the hood for centrifugation steps, and this further minimizes cell loss or the possibility of contamination. Additionally, the fact that the T cells follow a standard expansion profile shows that the Mars magnetic separation does not stimulate the cells, causing exhaustion. Continuing experiments are ongoing to confirm that the cells are not stimulated by positive selection in the Mars system. To summarize, Mars workflows are typically faster and less tedious than standard centrifugation or FICOL workflows. Automation or walk-away is a key benefit of the Mars system as it relieves lab personnel manual operation and hands-on prep time, allowing your researchers to focus on other assays as the Mars does the work for you. And at this time, we will consider any questions you may have. Sheila?
Thank you so much, Greg and Nick. We do have some questions that have come in. Okay, the first question, can you only sort one kind of cell each time?
The quick answer to that is no, because the data I showed on the CD4, 14, CD15 separation, you can do two cell types at the same time. And you can get the positive and the negative if you so desire.
Thank you. Next question. What is the maximum blood volume that can be used for CTC isolation?
Yeah, I can take that one, Sheila.
Sure.
So the way that our Mars wash is configured, we have tube holders that accommodate any tube size up to about 50 mil, a 50 mil conical tube. So if lysine is needed, as if you're processing whole blood, the maximum volume that we've really tested extensively is 10 milliliters of whole blood with adding lysine solution to bring that up to about 50 or 60 mils. However, with apheresis or bone marrow samples, no lysine solution is needed, and this allows you to process up to 50 milliliters of original blood samples.
Okay, thank you. Next question, I think this will be for you, Greg. 1 mil per minute. 1 mil per minute is a high flow rate. Shear force introduced, ? What is the volume requirement for sample?
So we request a minimum sample volume of 1 ml. Now that does not require 1 ml of whole blood because we usually go through a lysis process, and that is usually a 1:5 ratio. So basically, to run it through the chip, we would like 1 milliliter of fluid going in, whether that's lysed, with the lysing buffer. The quick answer to your question about shear forces, it is relatively quick, but we have observed, especially with the granulocytes, we don't see any clumping, which would actually be seen in our system after analysis on a flow cytometer. So again, as I said, we are looking at the activation of these cells using specific biomarkers. However, from what we see in our downstream applications, especially the flow cytometer, we do not think we're getting any significant activation of the cells.
Okay. How do you clean the system, especially for downstream molecular analysis?
That's a great question. And so basically, we actually have our own cleaning solution that we use, and we typically will run the cleaning solution through our entire microfluidics tubing between samples. That's all pre-programmed within the Mars system. And especially, to even take that one step further, if you're really working with precious and sensitive samples, we actually have replacement tubing in which you can replace the entire microfluidics tubing within the system, and it only takes about a minute or two to do total time, and it comes at extremely low cost. So based on your needs and really the sensitivity of your samples, we definitely provide solutions as far as ensuring that there's no residual leftover debris or anything left in the tubes before analyzing your next sample.
Next question. How much leukocyte can run to isolate CTC? Instead of whole blood, like 1 million cells or billion cells per run?
Per run, yes, you can run a lot of cells, but the key part is, as you heard from Nick earlier, we generally run around 10 mLs at a time. So it's not the absolute number of cells going through, as it's actually the density of the cells, which is why we ask you to, when we do the lyes, it's a 1:5 dilution. So you dilute the cells down to a certain point that it doesn't overwhelm the acoustic chip. But the absolute numbers, you're going to get a higher volume, but it's the volume, the cells per mil, that is the actual issue.
Okay, another question. You said you can go as low as 10 microliters to get 80% recovery. Can you go lower than 10 microliters?
Yeah, again, if you go lower, you're going to get actually a very low number of cells. And I think 10 microliters is the lowest we've actually used. To be perfectly honest, when we interact with customers, going lower is not usually the issue. It's actually going higher. But 10 microliters is the lowest we've gone.
Okay. What pore size of filter do you use?
We actually do not use filters. We do occasionally strain the blood through a typical blood strainer to get rid of coagulants and clumping, but we do not actually use a filter.
Okay, thank you, Greg. That looks to be our last question, unless anybody had any others. Oh, I'm sorry. I do see you have filter clumps before staining. Or do you have filter clumps before staining?
Generally, no. From what I've seen of the system, as I said, when we do the red blood cell lysis, we actually run those through a strainer, and we see very few clumps of cells coming out after the acoustic module.
Okay, so the cells are strained before running through Mars to ensure that you don't have clumping.
Right.
Yeah.
If there's any residual, clotting factors or things like that, because that will indeed clog the system.
Okay, thank you. Okay, so that looks like all the questions for today. You can always come visit us at appliedcells.com, and we'd be happy to answer any additional questions that you may have. Thank you so much for attending our webinar.